Okay, so in this lab, we will practice the sequence alignment and some basic ChiP-Seq data analysis. So if you go to this folder and you type ls, you're gonna see there's a zip file which contains all the necessary files for this lab. So the first thing we need to do is to unzip it. So just in the terminal, you type unzip, then the file name uab_ChiP-Seq_lab.zip. So see some files were decompressed from this file. So it includes 3 BAM files, which are the ChiP-Seq align the ChiP-Seq data, and the 2 FA files, that's the FASTA files used for alignment practice. So there's a document called uab_ChiP-Seq_lab.doc, which contains some description of this lab. If you open it, so I have it open here. Here are some basic instructions. So the purpose of this lab is to get students familiar with the usage of some popular software for analyzing second-generation sequencing data, especially the ChiP-Seq data. So the software, including bowtie, which is used for sequence alignment, R and bioconductor with a few packages. This will be used for some R-based practice. Then integrated genome viewer, or IGV, is used to visualize the data. So we have all these things set up in our true machine. If you are to use your laptop, you need to install this software. You have to Google, find the instruction for the software installation. Okay. So for the data, we use following data. The first one include the reference genome and the sequence read file for type of bacterial phage. So this will be used for sequence alignment using bowtie. The good thing about this data set is the reference genome for the phage is very small, only 5,000 plus base pairs. Okay, so because it has a small reference genome and a small sequence read file, the alignment is very quick. So the second data set contains the ChiP-Seq data for 2 DNA binding proteins, CMYC and PO2, and the control data. These are from a cell line called K562. So I have done the sequence alignment and generate the BAM file, but I only process the file to keep the reads aligned to chromosome 22, so that's only a small part of the genome. So the small data set makes the practice very quick. Okay, so let's start. The first one is to do the sequence alignment using the phage data, and we will use bowtie to do this. So there are 2 steps. The first step is to create index file based on the reference genome. The only thing you need to do is to run this command bowtie build. So you do bowtie build, then you provide the FA FASTA files for the phage. That's the reference genome. Then you provide a name. That's the name for the bowtie index. So if you run this command in the terminal, you can just copy and paste. Then you see bowtie output some information here. Basically, it built the reference genome. If you see the output, you see a whole bunch of EBWT files generated. Those are the bowtie index file used for alignment. If you are using some model organisms like human or mouse or a lot of other things, you don't have to do this step, because if you go to bowtie website, you see they're here pre-built indices. So they have all these pre-built indices for human, under different genome build for mouse, rat, cow, and so on. There are a lot of them. So if you are working on using these organisms, you don't have to do this bowtie build step. You can just download from here. Okay. But since I'm using phage and bowtie doesn't have the pre-built index, I have to do this step. So once you have the bowtie index built, you can just run this bowtie command to do the alignment. So this particular command I'm using here, -v3, -f, -s, and so on, indicate I want to do the alignment, allowing 3 mismatches. And I want to output the result in a file called reads.sam. That's the output. This part is the input. This part is the index file I just built from this line. And the output will be a format called SAM. There are different alignment data format, and SAM is one of the most popular ones. Okay, so I just run this command. I can just copy and paste to the terminal. So it's very quick, just finished in no time. So it tells me 77.7% of the reads can be aligned, 22.3% cannot be aligned. Okay, so this might give you some impression that the alignment is quick, but remember we are aligning a very small genome, only 5,000 plus base pairs. If you do this alignment in a real, like a human genome, it usually takes much longer. It takes maybe, depends on the number of reads you have, it usually takes half hour to several hours. So that's all about the sequence alignment. There are some other things I didn't include in this lab. For example, you can use a tool called SAMTools to convert the result SAM file. Let me show you the result. Is here, the reads.sam. This is a SAM file. SAM file is a text file. So this is a text file. This is the result. Okay. So if you can use SAMTools to convert a SAM file into a BAM file, which is a binary version of the SAM. The size of the BAM file is much smaller. So we usually do the alignment, generate the SAM file, then convert it into BAM to save some disk space. Okay, let's proceed to the next topic, the ChiP-Seq peak calling. So I covered the max in the ChiP-Seq class. Okay, so here I'm going to use max 1.4 to call peaks from these two files, or for these two proteins, CMYC and PO2. So to use this is very simple. Just use a command like this. It's max 14 minus T. This is the treatment. So this is basically the IP sample. You provide a BAM file. Minus C means control. So here is the control file. Minus N means the output file name. So I call it K562_CMYC. So if I run this command, I can just copy and paste them into the terminal. So Max outputs some information here, so it's running. It takes a little bit of time. So for a full file with 20 million reads, it might take 5 to 10 minutes. See here, because I'm only keeping the reads from chromosome 22, so it's much smaller. It's finished in a few seconds. So if you look at the results, it generates several new files called K562_CMYC_peaks. There's a peak list. There's the summit of the peak. There's some negative peaks, so you don't need to worry about this one. So if you open up this file, you can see here are the list of peaks. So for each row represent one peak. You have the chromosome name. This is chromosome 22. Start and end location. The length of the peak, the summit of the peak, and you have this is the last column is the FDR. This is the fold enrichment. Here are the -10 times log 10 p values, and so on. Okay, you can see there are the list of peaks. So I can run the same thing for PO2. Let me quit from this file. I can run this for PO2. Okay, now I also have a list of PO2 peak files. Okay, so that's it for peak calling. It's very simple. Next, I want to introduce this tool called IGV, Integrated Genome Viewer. I think it's a very powerful and useful tool for visualizing the data. I use this a lot. It's developed by the Broad Institute, and it runs on all operating systems. So if I start IGV here, I can do igv.igv.sh. I can start it. Okay, it takes a little time, depends on the network speed, because it needs to load some data from the internet. Okay, let me run this. So it's a Java program. So loading the genome from the internet. So here's IGV. Let me make it slightly bigger. Okay, so IGV can be used to visualize all kinds of sequencing data, including DNA-seq, RNA-seq, ChiP-Seq. So in here, I want to change the genome build because the bind file I got was aligned against the human HG18 genome, so I need to change it to HG18. Now I want to visualize my ChiP-Seq files, but the ChiP-Seq are the raw data are the bind files, including the information for all the reads. But if you visualize the ChiP-Seq, you don't need the reads. You only need the bin counts. You summarize the counts, number of reads into the equal-sized bins. So to do that, you first need to convert the bind file into something called TDF. It has an internal format, basically summarize the bin counts. So to do that, I can run IGV tools. I just click tools. There's a run IGV tools. So it pop up this window. So the command is count. I select input file. Then I go to my folder. Then I can take this CMYK BAM files. .bam I select. So this is my file. The output will be a TDF. Just add a TDF there. Then I click run. It's done. So it's very quick. This is CMYK. Then I'll do the same thing for port 2. I click run. Okay, now I can close this. You look at your folder, then you have 2 TDF files now. Okay, so you can load the TDF into IGV to visualize them. You can do it from file, load from file. I can select just 2 TDF files and click open. Now the file was loaded into IGV. You can see the IGV visualize the data from this whole genome from chromosome 1 to chromosome Y. But since I only have the reads for chromosome 22, so only here has some read, I can just double click them. So here are the data. So this is showing the whole chromosome 22, so it's way zoomed out. So now I can zoom in. I can just double click, or I can do it from here. Then you can see here are the bin counts. So basically shows at this location there are this many counts. I can change the range of the data. Basically here are showing the y-axis limit from 0 to 2.39 or something. So I can select these 2 tracks on this panel, right click, then I can set data range. I can make the max to be like 20, for example. Okay, then see these bars become much lower. Now I can zoom out. So you can zoom in some places to visualize the peaks. See the peaks here? So I can zoom in. Here is a big peak. Okay. So you can drag this left and right. So IGV is very useful. I spend a lot of time on IGV. Sometimes I load in like 20 tracks, and I zoom in, zoom out, drag left and right to visualize my data. A lot of times you can see patterns by just looking at the data. For example, for these 2 data sets, CMYK and PO2, when you look at them, you get an idea. You can see the peaks have very good agreement. Okay, there are 2 binding proteins, but they kind of occur at the same locations, right? So you see whenever I see a peak in CMYK, you see a peak in PO2. This kind of relationship can be visualized in IGV. Now I will switch to get into R to do some data analysis. So here I'm gonna use R to do some comparison of the peaks. Okay, so basically I have two lists of peaks from CMYK and PO2. A simple thing I want to do is to look at the overlap of the peaks, because we just visualized on IGV. It seems like these two peaks often occur at the same location. So to do the overlapping analysis, you can use other tools like BED tools, but I usually do it in R using a package called dynamic ranges, which is very powerful and useful. Okay, so I have this R code in this Word document. I also have it. It's called the quicktest.r as an R file. Okay, so now I'm switching to RStudio, and all the R codes are here. You can run them in R. So let me select a few lines to run. So the first thing I'm going to do is to read in the peak files. So here are the peak files generated from Max. Okay, so I read them in using this read.table function. I run them. So here are the peaks I just read in. Next, because the peaks are read in as data frames, but the dynamic range package uses a special object called JRanges to do the comparison. So I need to create the JRanges object from the peaks. So basically I call the JRanges command. Now of course it has some special requirement. I don't have time to cover this. You have to read the manual to learn how to use this thing. But basically I can do this. I run these two lines. Now I got these two objects, pics.po2.gr and pics.cmyk.gr. If you take a look at these two things, run. So here are the pics.po2.gr. It's a JRanges objects with 6 ranges. Here are each line is a peak basically. This is Po2. This is CMYK. Okay. Now, if I want to look at the overlap of these two lists of peaks, I can simply call this command over. This line basically means for each peaks in Po2, whether it's overlap any peaks in CMYK. So this one will give me a list of logical vectors, true and false. So then I take a mean of this list of true and falses. Then I can have the percentage. I can hit run. So this one basically tells me 57% of the Po2 peaks overlap CMYK peaks. If I run the other way, I got 62%. So basically 62% of the Po2 peaks overlap CMYK peaks. Now let's go to the next step. So this is an optional practice. It requires some kind of advanced skills. So I basically want to look at the aggregate bonding signals around the transcriptional starting site. So this is an analysis often used in many genomic papers. People look at different things around different places. So the idea here is that I want to look at the transcriptional starting site across all the genes. Then I want to average the signals to see whether there's a common pattern there. So basically it's the average signal around the TSS. So this analysis can be done by first obtaining read counts in equal size bins around the OTSS. Then I compute average. Okay, so the code below basically shows how to do that. First of all, we need to get the location of the genes and the location of the transcriptional starting site. Then we create equal sized bins around all the TSS. Then we obtain the read counts within all these bins. So I get a count matrix, then I take an average. In this Word document, I have reasonably detailed instruction. But here I'm just gonna run this in R. So first, I use a library called genomic features to extract the gene information. So genomic features is very useful for that. It can basically, if you do this, make transcript DB from UCSC, you can download and process the gene information for a particular genome. In this particular line, I got the REF-seq gene for HG18. So this basically replaces the step where you have to go to UCSC, go to the table browser, and download and process within the file. Okay, so here I'm not running that line, this make transcript DB line, because it takes usually 5 to 10 minutes. But you only need to run this line once, then you can save the result. Then in future analysis, you can just load them in. So in here, I load in genomic features and load in this file. Now here is my pre-saved TXDB. You can see this is a TXDB object. It's from UCSC. It's on HG18, and so on. Okay, so this is basically a little database storing all the gene informations. Now I can take all the transcripts. Then I can take all the transcripts on chromosome 22, because remember the dataset I provided is only on chromosome 22. So I run these lines. So I get my tx.chromosome 22. So I got 687 transcripts on chromosome 22. Then I obtain the transcriptional starting sites. So it's basically the starting location of all the genes or all the transcript. I don't have time to explain all the details, but basically you use this start location of all the genes, but you have to pay attention to the strand direction, because if it is the minus strand, the end location is actually the TSS. If I run it, I got my TSS. So it looks like this TSS. They're all the genome locations. Remember, these are all on chromosome 22. Here are the transcriptional starting site location for my 600 something transcripts. Then the next few lines is to create 50 base pair bins around the TSS. Okay. So basically take upstream and downstream 1,000 base pairs, so it's 2,000 base pairs centered at the TSS. Then come into equal-sized bins of 50 base pairs. Okay, so I run this. So I got all the bins as G range, so they're windows. Next, I use a library called rsam2s to read in the SAM file. So basically, there's a function called scan BAM to read in the BAM file. I can read them in. Then for all the sequence reads, but after I read it in, for all the sequence reads, I create the G-range object. So each read is represented as a genomic interval on the genome. Let me run this. So if you look at this, this is a G-range object with 113,000 ranges. So basically, this is the number of reads. Okay, so each read is represented as an interval. So now I have all the TSS, the bin window around TSS as the genomic intervals, and I have all the sequence reads as genomic intervals. I can do a count overlap here. So basically, what this line does is to count for each window in this list how many reads fall into each window. So I can do this counting really quickly using this function. Then I do this. I put it into a matrix. So I got something like this. So this is a matrix. Of 687 rows and 40 columns. So each row represents one transcript, and each column is a window. Remember, I have 687 transcripts. For each transcript around the TSS, I create 40 dynamic windows. Now I can just take a min of this thing, do a column min, and plot it. So if you practice this, you might want to comment out this PNG line and the dev.off line, because these two lines will output the figure into a PNG file. So if you just plot it, you see something like this. Okay, so basically here is the TSS. Here are the 20 windows downstream and upstream. So basically you can see that around the TSS, the average bonding signal looks like this. Okay, so the bonding is the strongest at the TSS. Okay, so that's all for this lab. So basically I taught you how to use bowtie to do sequence alignment, how to use max to call peaks. After you have the peaks, how do you visualize them in IGV, and then how to do some overlapping analysis and some other analysis using R.